to talk about transforming principle. Transforming principle is one of the most important topics for class 12th. It comes under molecular biology, that's right after genetics. This was a very trademark experiment, a very benchmark experiment, because this was kind of like the starting point which helped us to find out what the genetic material is. They had a wide variety of options. They had an options that there could be proteins as the genetic material, or DNA, or RNA. These experiments started to find out which of these was actually the genetic material that actually helps in passing information from one generation to the next generation. So this experiment as conducted was known as the transformation experiment. Transformation, because here one cell type got transformed into the other, and that is what we are going to see here in the transformation experiment. Just to give you some details about it, about the person who was the first to conduct this experiment. So this experiment was conducted by Frederick Griffith in the year 1928. So he was the first one to conduct this experiment. He used a particular bacteria. That bacteria is known as Streptococcus pneumoniae. Streptococcus pneumoniae is the name of the bacteria over which this experiment was conducted. And now we are going to move forward with the details of the experiment. This experiment was very important. It was the first which helped us to identify the nature of the genetic material, that whether it is DNA, RNA, or proteins. So let's come to the main part of the experiment now. There were two strains of this bacteria that were used. One was the S strain and the other was the R strain. Coming to the details. The S strain, as the name is based on, is smooth. This is the smooth colony forming bacteria. So इनकी जो colonies होती हैं, they are very smooth walled. You know, the walls of these bacteria are highly smooth, and as a result उस bacteria की colony भी extremely smooth बनती है. Here's the nucleus for each of them. On the other hand, R stands for rough. So the R strain bacteria forms a rough colony. This is how you can physically distinguish between these two types of strains. So this is the kind of colony that you see for the R strain. This is one bacteria, two, and obviously a colony has many bacteria. Okay, but a very common thing that said is जो जैसा दिखता है वो वैसा होता नहीं है. So out of these two, the smooth strain is actually the virulent strain. Virulent means that it is capable of causing infections. On the other hand, the R strain is known to be the non-virulent strain. So this is the one which is non-infective. It does not cause any infections. The infection caused by the virulent strain is actually pneumonia, as you can all make out from the name only. It causes pneumonia in mice. And this difference between these two strains was what was used by Griffith to find out what is the nature of the genetic material. So he conducted this basic experiments in 4 steps, and I'm going to highlight those 4 steps for you here. So first, he took a healthy mouse. He injected the S strain of Streptococcus pneumonia into this mouse. So he put in S strain. What do you think the output was? Obviously this is the virulent strain. Mice got infected and the mouse dies. So the mouse died at the end of this first part of the experiment. Second step, another basic thing, he again took the mouse. This time he injected the mouse with the R strain of the bacteria. R strain, that is the non-virulent strain. Clearly this strain will not be causing any infection and the mouse will live. Right. Now the third part of this experiment is where the information or the outcomes began. Again he took a mouse. This time he inserted S strain, but in a different way. This time he inserted heat killed S strain. He killed the S strain bacteria first. Heat killed S strain. This time he saw that क्योंकि उसने बैक्टीरिया को मार दिया था because of heat treatment, it could not cause infection, and as a result the mouse lived. So heat killed S strain, very clear to us, is not capable of causing infection inside the mouse. Fourth part of his experiment revealed the true thing, and he found out that in this part he took heat killed S strain, again heat killed S strain. I'll just mention this in the bracket. In addition to the heat killed S strain, he also took the R strain. He kind of like made a mixture. So if we come to think individually, heat killed S strain is not capable of causing infection. R strain by itself is not capable of causing infection. So the mouse should have lived in this part of the experiment. But as it turns out, the mice died in this part. Now, how did this happen? This is where the main thing for molecular biology was worked out. This combination of heat killed S strain and R strain. This is where Griffith found out that this combination is infective. He clearly made out that there is something which transformed the R strain into the S strain. So that was the transforming principle. The transforming principle, what did it do? It changed the R strain into the S strain. Forgot to write S, my bad. Yeah. So this is what happened. Now everything relies on this transforming principle. This thing that was responsible for this transformation. The nature of it was not identified by Griffith, that whether this transforming principle was DNA or it was RNA, or it could have also been proteins, because that is also another major information storing biomolecule. This nature was not found by Griffith. He only identified that there was some transforming principle. It was identified to be DNA later on by some other scientist going by the name of Avery, MacLeod, and McCarthy. These were the three scientists that later on, using some other set of experiments, found out that DNA was the actual transforming principle. But Griffiths experiment proves as the starting point. It was so very important. It was the starting point for finding out what a transforming principle is, what its work is. Later on, the nature was being identified by three other scientists. This is a very big discovery. It's very important for the field of molecular biology. Just to give you a graphical representation, I'm going to add an image here. Yeah, so we have this image now. This just gives us an idea of how it's working out here. So if you see in the first part, there was a rough strain. As you can all see, it's non-virulent. When it got injected into the mice lived, right? After which, S strain, you can see the difference in the colonies. They have this smooth polysaccharide coat over them. This is the smooth polysaccharide coat, why they are named S strain. And this is the virulent strain. When it got injected into the mouse dies. Now, you have the heat-killed S strain. They killed it using heat. It lost its infectivity, and as a result, the mouse lived. Finally, they took a mixture. They took a mixture of the heat-killed smooth strain and the rough strain. Individually, neither of them could cause the infection, but when they came together, there was something that transformed the rough strain into the smooth strain, and as a result, we saw that the mouse dies. This is what Griffith's transformation experiment was all about. I hope you all got a brief idea, and this is a very important topic from the point of view of boards or from the point of view of entrance examinations like NEET. And yeah, all the best to everyone. Stay tuned for more videos.